WACCBIP. He's a senior research fellow here, and he has been coordinating this project. And he would start off with the seroprevalence a bit. By the way, colleagues, there are people in the waiting room. I see people in the waiting room. I don't know who's smiling. Please let people in. So Peter, over to you. Thank you.
Thank you, Prof. Good afternoon, colleagues. I'm going to start my video. Good afternoon, colleagues. My name is Dr. Peter Kwashi. I'm a senior research fellow and deputy director in charge of research here at WACCBIP. And I'm going to just take you through sort of a snapshot of the seroprevalence work that we've been doing across Ghana, Nigeria, and Burkina Faso as part of this project. And I won't talk too much about our partners because Prof. Awandari has already talked about them quite a bit. One that's not part of the WANIDA network is the Group de Recherche Action en Santé. That's one of our Burkina Faso partners, and they do research on scientific issues in Burkina Faso, and so they're a great partner of this. And this project, mainly across the three partners, was funded by the Rockefeller Foundation. And for the seroprevalence work, also later on in Ghana, we have the FCDO, which I'll get to in a bit. So our study sites, for those who don't know West Africa, we are here. This is, we are here. This is Ghana. Burkina Faso is right above us. Nigeria is to the right, separated by two countries. But there's suspected extensive cross-border talk, even when the borders are closed. So there is, and also Nigeria is the most populous country in Africa. So it's good that while we are doing our seroprevalence work, we include such a populous neighbor. And if you look in terms of confirmed cases, why is this not working? In terms of confirmed cases, in Ghana, as at yesterday, we had about 161,000 cases with 1,445 deaths. Nigeria has 256,000 cases with around 3,000 deaths. Burkina Faso is much less, 21,000 cases, about 384 deaths. You know, so one thing that will also be interesting is whether the seropositivity that we see correlates with the case reports, and this is what in terms of confirmed cases, it looks like. This is from the WHO dashboard for Africa. And our first manuscript that Prof. Awandari mentioned covered early outbreak in Ghana. That's from all the way from March till June. Our first seroprevalence work, which was done mostly in Greater Accra region and some selected regions of Ghana, was from, I guess you would say, the middle of the pandemic, of the middle of the Ghanaian outbreak. So it covered the tail end of wave 1 and most of wave 2. And this project, basically we've covered from wave 3, 4, and it looks like we'll continue covering 5. And the whole aim of this project was to extend the seroprevalence work we've been doing in Ghana, and also the sequencing work we've been doing in Ghana, and to start some of this work in our neighboring countries. And Professor Andari talked about the cases that are starting to rise here in Ghana. So in Nigeria and Burkina Faso, basically the sampling periods for the seroprevalence work is what I've indicated, from September all the way through to now. So the wave stratification is based on the WHO platform, like I mentioned, Dashboard for Africa. And wave 1, we classify as at April 2020 to August 2020. Wave 2 is November to May. Wave 3, June 21st to November 21st. And wave 4, December 2020 to May 2022. Okay. And the study was structured such that because we have a seroprevalence arm and a sequencing arm, we had a broad, somewhat broad ethical protocol. And participants, when we go into the field, participants had informed consent if they agreed to take part in the study. There was a questionnaire administered. In the field, antibody test was administered, sometimes antigen test was administered. A positive antigen test would result in PCR being done, and that would be sent for next generation sequencing, which Collins will present later, results from that. Also, we get some samples from the testing facilities which have adequate metadata. They're sequenced, the analysis is done, and then they deposited in GISAID. So what I'll be talking about mostly is from consent through the antibody test. So before we do anything, we had to train our field teams on our protocol. We were using a rapid diagnostic antibody test, which we got in Ghana. We had permission from the FDA to use for the field research. And in Nigeria and Burkina Faso, those were already approved by their respective FDA for their field research. So we had trainings both in Ghana and our respective partners. And it's not moving. And this is just some pictures showing the ongoing work in the field. We targeted malls, markets, institutions, religious settings, health centers, community areas. And of course, that differs slightly from site to site. So this is at ACE-GID, ACE-FAP, GRASS, eTech-MTV. And then let's dive into the data. So if you look at Ghana, I mentioned earlier that we had done some work previously in Ghana, and at the end of that period in April 2021, we had seen seropositivity rise from around 19% at the beginning of that work to 25%. So from June to, this summarizes the data in Ghana from June to November 2020, end of November 2020. And you can see that our seropositivity ranges from as low as 27.3% and 7%. And it also makes sense that right near the top end of that seropositivity, Greater Accra region, which is the epicenter of the Ghanaian outbreak, had about 55% seropositivity. Western region, which is our third most populous region, but also has significant influx of foreigners due to the oil industry there also had a high percentage of positivity. And Ashanti region, which is another populous region and which is the second largest outbreak in Ghana, also had a high positivity. If you, and this is sort of looking down at Greater Accra in more detail. This first bar is a summary of our earlier work, where on average it was about 19% seropositivity, and that coincided with the Delta AD614G and B1 variants. So these were all the early variants. And then even during the alpha wave, which in Ghana was not just the alpha wave, in the northern part of Ghana, we also had ETA and we had A27. And so the positivity went up slightly, but it was still in the 20s. When we went back from June to November, you saw that the positivity had gone on average to about 55%, which is indicative of the Delta wave. And currently we're in the Omicron or post-Omicron period, and the average positivity here is in the 70%, so 70% range in Greater Accra. And this is basically mirrored if you expand to the country, right? But when you actually dig into the data a bit deeper and you go by month across Ghana, you see that as the rate rises with Omicron, that's from December to January, you have 80% and above seropositivity. In April, it actually dips, and this is not, the data here is not across the country, but in the major centers, that is Accra and Kumasi. And I showed you earlier that Accra mirrors quite well how the whole overall outbreak in Ghana looks. You see a dip in positivity. And what that we think that is if you look at the waves of infection, there was, after Omicron, there was a high during Omicron, there was a high number of infections over a short period of time. What we believe is that basically killed off the local outbreak. And so there were no reinfections which were happening, and the people lost their antibody expression. And that's what we are seeing from April through to May. And then you go to Burkina Faso. So prior to this, as far as we know, there wasn't data available for Burkina Faso, and we had the two centers, ETEC NTV based out of Ouagadougou and GRASS based out of Bobo Dioulasso. And they had seropositivity ranging from 16.6% to 33.5%. And this goes back to what I was talking about, that Burkina Faso had a much lower confirmed case count than Ghana and Nigeria. So it actually makes sense that their positivity would be lower than what we're reporting. Why the discrepancy between the two places is not quite clear. Also, Burkina Faso is more landlocked compared to Ghana and Nigeria, and there's a bit of instability. So it's conceivable that air traffic was not or travelers entering the country was not as high as in Ghana and Nigeria, particularly post-lockdown. And then if you dig into the data a little bit, you can see that even though the epi curve was relatively flat in October and November, you had high seropositivity here. And that went down again. And we didn't sample during December in Burkina Faso, so we don't quite know whether we had a high peak in we don't quite know whether we had a high peak during Omicron, but you can see that this dip that we're seeing in Ghana is also reflected in that data. And switching over to Nigeria, our two sites were ACEFAP, which was in the north in Kano, and ACEJIT, which was in the south. And we basically covered 4 states in Nigeria, and we saw that the seropositivity in Nigeria was very similar to Ghana, quite high during the Delta wave, and overall seropositivity of 47.7%. And this was a bit interesting because the rate was highest during Delta, and then it sort of tapered off for a while. And this is data that we're now still collecting in Nigeria, and it's quite high in May. Quite sure why that is, but that's also possibly because of the large, Nigeria had many sub-waves for different regions compared to Ghana. So when you compare the data of the 3 countries together, you see that overall seropositivity is lower in, not quite half, but lower in Burkina Faso right from the start of the project. And that's consistent. Now it's sort of a bit higher than Nigeria in the post-Omicron or I guess you can say pan-Omicron era, or because now we are in multiple variants of Omicron. So I don't know if we still call it Omicron era. And Ghana has basically has gone up to 80%. And yeah, and if you dig into the different demographics and risk exposures, you see that strangely, and this we've seen in also Nigeria, females had a higher than normal risk of being positive compared to males, which was different from our early data sets where the males were the most seropositive. And also age is positively correlated with seropositivity, which is has been reported everywhere. And that's also, we see that in the Burkina Faso data. We see that higher level of education and higher income in Ghana is correlated with increased risk of seropositivity. And this is consistent in both Ghana, Burkina, and you see the same trend in Nigeria. Of course, I can't really go that deep into the data with the time we have here, but digging a little bit deeper, if you look at those who are seropositive, how many of them have had a PCR test? You see that right at the beginning, about in Accra, about 5.9% of them had a PCR test, and that went up to 14.9, and in 2022, that's 8.9. So that tells you that most people did not know that they had COVID-19. And that is something that cuts across all our countries. 8.7% of people in Burkina Faso who were seropositive had a PCR test, 6.8 in Nigeria, and 11.9 overall in Ghana. If you look at whether could vaccination explain this seropositivity rises that we're seeing, you look at the 2021 data in our study population. In Burkina Faso, only 3.7% of them had at least one dose of vaccine. In Ghana, it's a bit higher, 26.6. Nigeria is also low, 7.4. So then you can't really, and then you look at the 2022 data, it's gone up in Burkina Faso, slightly up in Ghana, and it's actually gone down in Nigeria. So then that tells you that these increases that we're seeing are not linked to vaccination. They are linked to natural infection. So I'll summarize the sero-epidemiology study. We saw high levels of SARS-CoV-2 seropositivity across all study areas. The reason I'm using seropositivity is because it's not necessarily an indicator of prevalence if you are losing your antibodies, right? So it's seropositivity across all study areas, which indicates that you have undetected transmissions, large-scale undetected transmission across all these countries. And we could see that these were linked, the increases in seropositivity was linked to infection waves, as can be expected. Certain locations and demographics had elevated risks of exposure, particularly religious settings, institutions, where people tend to be in enclosed areas in close proximity. And we saw that large infection waves were followed by low transmission periods. And also that likely that means that high population exposure probably limits new local transmissions. And that's something that seems to be continuing in this Omicron / post-Omicron era. So far in Nigeria and Burkina Faso, we don't see increases in the reported cases. And I mentioned earlier that the Burkina Faso seropositivity appears to agree with the smaller outbreak. And this is something that will be putting out a detailed analysis very soon, so stay tuned. I would like to acknowledge that in Ghana, we worked with quite a few partners, and that's why we were able to have a large number of samples. That's through the facilities of the Ghana Health Service, such as Navrongo Health Research Center in Navrongo, the Christian Health Association of Ghana. Our early work was aided by some of our collaborators at Noguchi and UGMC. And we also had some partners in University of Health and Allied Medicine in Volta region, CKT University of Science and Technology, KNUST, that's the Kwame Nkrumah University of Science and Technology, and University of Development Studies. And our field team, I have to acknowledge our field team led by Dr. Daniel Drew-Mensah and Dr. Gloria Migacha, and our data manager, Francis Jabing, who basically aggregates the data from all the countries, all the sites, and does the analysis. And of course, the WACCBIP COVID-19 team. And I mentioned that our work we have many funders for our work. Most of what I presented here was funded by Rockefeller, with some of the 2022 data being funded by FCDO. This work, of course, wouldn't exist without the World Bank grant and the African Centers of Excellence. And our early work in 2020 was funded by TIBA, Tackling Infections to Benefit Africa. Thank you very much.
All right. Thank you very much, Peter. Great. We're going to move straight to Collins. So Collins Misita is a postdoc at WACCBIP, and he is the bioinformatician working on our SARS-CoV-2 genome sequencing project. So Collins, please share your screen and take it away.
Thank you very much. My name is Collins Misita Moranga. I'm going to talk to you about the genetic epidemiology of SARS-CoV-2 in Ghana and Burkina Faso. And just to start off, I think all around the world, we've seen different waves of SARS-CoV-2 infections. And you can see that in Africa, we've had a fair share of different waves going around, following the waves across the world. And you can see that way earlier in 2020, we had the first wave, which was basically characterized by, some of the variants that were causing serious infections. And then the second wave around January 2021, which was characterized basically mostly by the Alpha variant. And this was followed later by the Delta wave that caused significant infections. And we had a final wave towards the end of last year across Africa. And now we are seeing a new wave with a new Delta Omicron variants. And you can see that in Ghana, I think the number of cases has always been a little bit low compared to other parts of the world. But still, we've had our fair share of different waves, surges, although there has always been a lag behind the cases in Ghana. And since February, March, thereabout, the curve had flattened for new cases. But we are seeing a surge of new cases, and I think this was yesterday. We had about 46 new cases and 396 active cases in Ghana. So that means we're experiencing a new waves, and I'm going to talk to you about, what are the new variants that are causing these new cases that are going around the country. So just to start off, what we do is basically collect samples from different parts of the country, and we collect different metadata information about the patients, mostly the age of the patient, the travel history, and the location where the sample is being collected from. And we process this through the ACTIC protocols for sequencing, and we analyze the genomes using still the ACTIC platform. The initial samples, we did them with using MySeq, but most of our genomes, we've processed them using MinION platform. We follow this with phylogenetic analysis and different types of analysis, and this includes determining which are the new circulating variants, what is the distribution of the variants across the different regions in Ghana. And we also look at what are the trends, how the variants are changing in terms of the genetic diversity of the variants, what new mutations they're acquiring, as well as look at the phylogenetic relationships between these different variants. So just to start off is that over the past, since 2020, we've sequenced and analyzed about 2,100 samples across 3 different countries. And Ghana we did most of the samples from Ghana, and this is about 2,000 samples. And we've also sequenced some samples from Nigeria, 42 samples, and some samples from Burkina Faso. And in Ghana, mostly we've tried to collect and sequence samples from different parts or different regions, and these include the Upper West, the Savannah region, the Bono region, Hafo, Shanti, Central, Western, and as well as, the Upper East. But most of our samples have come from the Greater Accra, as well as the Central region and the Ashanti region, which are, the highly populated areas. We've also collected samples from travelers arriving in the country, and this was samples that are arriving at the Kotoka International Airport. And this was basically to try and track which variants are being imported into the country throughout the time. And so just to start in terms of results with the Burkina Faso samples, the 15 samples we got, these were samples that were collected mostly between September and October 2021. And most of the samples had the Delta variant. And what we saw in terms of clinical information is that most of these individuals had symptomatic infections. And the age was varying between 19 to 57 years old. And they were both Delta and Delta plus subvariants that were being characterized in these samples that were got from Burkina Faso. And from Nigeria, we got samples ranging from December 2020, January 2021, all the way to January 2022. Although it's 42 samples, but what we saw is that in these particular samples, the Eta variant was dominating, followed by the L3 variant. And now the samples that were sequenced from October 2021, we saw a surge in Omicron cases, Omicron variants. And to move to Ghana, out of all the 22,000 samples that we've sequenced since 2020, we've seen that Delta variant is the most dominant variant in terms of accumulating about 500 samples, and followed by Omicron and the Alpha variant. And you can see that most of these high frequency variants were detected in some travelers, that is Alpha variant, the Eta variant, were detected in travelers that are arriving in the country. And is that most of the high frequency variants comprise about 71% of all the SARS-CoV-2 genomes that we've sequenced. And this is a high number compared to all the other variants that we've detected in the country since 2020. And this, we've seen more than 50 different variants throughout the time, but these particular variants of concern have remained dominant, and they've been characterized by those different waves that I talked about earlier. And those travelers who arrived in the country, when we check their travel history, or at least the reported countries of origin, most of these travelers came from different parts of the world, including from the US, the UK, and other African countries. And some of the samples that we sequenced at the time from travelers arriving in the country, we saw that some variants were being detected from travelers from other African countries, although those countries had not even reported those particular variants. And this was a demonstration of very good surveillance, and we could, in fact, deposit the genomes in GSAID and report for those countries as well. And if we look at the distribution of this SARS-CoV-2 by the different sampling locations within Ghana, and here I'm showing a plot whereby we have the percentage number of cases across the different regions. And then on the x-axis, we have the different regions across Ghana. We've tried to sequence samples from almost all the regions. And from what you can see is that there's a fair share of sharing, especially the regions that have very high population. That is the Ashanti region, the Greater Accra region. Although one, and that's why we labeled it a variant that was under monitoring. But then the detection of Delta variant in community samples around May 2021, we later saw that it took over all the infections, supplanted all the other different variants, and it was the only variant that was dominating across the country. And around November 2021, I think Omicron came in, and it has overtaken the number of cases that we are seeing. We sequenced a lot of samples in December 2021, and you can see that most of them, about 98% of the cases were basically Omicron variants. And when we look deeper into these different variants, what we could see is that this plot is showing the number of mutations in different variants, and you can see down here on the x-axis is the different variants. And you can see that, so each particular dot represents each particular sample and the number of mutations that particular sample had. And you can see that most of the samples, most of the lineages or variants that dominated in the country at any particular point in time have a wide range or diverse range of mutations. And this is characteristic of, you know, the local evolution of the variants that is happening within the country. And you can see that from A27 having a wide range of mutation ranging from 20 to about 30 mutations. A27 dominated in the country along the northern regions, but it was minimally detected down in the southern regions, Accra and Ashanti regions. And then you have the B1318 variant, which you can see that the different samples had different amounts of mutations in their genomes. And this could, you know, signify the differences in terms of transmissibility as well as, you know, severity of the infections. And then you have alpha variants and delta variant also having a high number of mutations, as well as the 525 eta variant. And you can see that in terms of the number of mutations, basically Omicron samples have the highest number of mutations, ranging all the way from 40 mutations to 60 mutations. And all these different samples, you can see there's kind of a mean number of mutations, which is around 50 mutations. So if we look at just the spike mutations in terms of sharing, you can see that all these different mutations that have already occurred in a spike mutation over time. And you can see that the Omicron, the Delta, the Iota, the Eta variants, most of these share a lot of mutations. And one of these particular mutations is the D6114G, which characterized most of the B lineage mutation and was responsible for initial severity of the COVID-19 pandemic. And you can see that in terms of the frequency, almost all the samples have the D6114G, and different mutations have a high frequency, and some of them are unique to a particular variant, and some of them are being shared across different variants of the COVID-19. And these mutations actually translate into the level of phylogenetic relationship that we see among the different variants. And you can see that, so this is a phylogenetic tree that is showing, you know, the divergence of the different variants across time, as well as the number of mutations each of them have. And you can see that Eta, Delta, Alpha variants, as well as the Omicron variants are the most diverged variants, and they account for most of our cases. And you can see that even within that particular lineage, let's say, for example, Omicron, you can see there's a lot of variation in terms of, although they have a relationship by being on the same particular branch, but there's a lot of variation between the different samples that we have. And you can see that recently there's emergence of emerging lineages, that is the BA5, BA11, and the BA4 lineage. And you can see that all the other Omicron lineages are on a separate branch, but then the BA5, the BA4 are characterized by branching or diverging through this particular node here. And what we see is that initially, during the initial infections, the BA1 particular subvariant of the Omicron dominated most of the Omicron infections in December, January, thereabout. But then in Ghana, we haven't had BA4 being detected in infections since they were reported. BA4 was reported around January 2022, and BA5 was reported around February 2022. And you can see that it is now that we've been able to detect the BA5, I think one sample in our infections. But then the BA1.1 subvariant, which has also been responsible for infections in different parts of the country and being dominant in terms of infection, we can see a fair share of this particular variant. So if we put the mutations as well as time, you can see that the Omicron variants have the highest mutational fitness, and this could infer any either transmissibilities. Right now, reports are saying that Omicron kind of has mutations that can increase severity of infection or supplant the other Omicron variants. And you can see that the BA4 is already supplanting the BA11 mutation, as well as the emergence of the BA5. But basically what we are showing is that we've had all these different mutations, all these different variants over time, but Omicron seems to be having a higher fitness level in terms of, and we've seen this through the high levels of transmission across different populations around the world. And in our samples, we are seeing that the same Omicron has a higher level of fitness compared to the other earlier variants that we had. Now, just to summarize all these particular disease trends, we are showing that, the earlier infections were basically dominated in December to May, were dominated by the Alpha variant, which was basically down in the Greater Accra regions, the Ashanti regions, and the northern regions were dominated by the Eta variant. And you can see there was kind of an interplay between the cases, the sequences we had for Eta as well as the Alpha variant. But then later, there was the emergence of the 318 variant, and this took over infections from Alpha and Eta and dominated infections for almost 3-4 months. In Ghana, we labeled this a variant that was under monitoring, and then the emergence of Delta took over all the infections, and it supplanted even all the other different variants until Omicron came up. Now, since then, Omicron has been high, and it has dominated infections since December 2021. And we just sequenced samples from May 2022. And you can see that these are samples that were collected between May 18th and 30th. And you can see that the BA.4 lineage is the most dominant in all these samples. And I've already mentioned that the BA.1 subvariant is an emerging lineage since January 2022, but then it hadn't been detected in Ghana till we've detected it. And you can see that the number of cases actually is very high, because most of these cases report to LEDIC, which is a site for infectious disease testing. And the other lineage that we saw in one sample was the BA.5, which also appears to be emerging. And other countries are saying that the BA.5 lineage is actually taking over other regions, other locations in terms of the number of infections. It's appearing on the news. So I think Ghana, we should also be concerned, because the BA.5 lineage seems to be supplanting the BA.1 lineage that were already circulating at the time. And our colleagues from South Africa, who've shown that, among unvaccinated people, the BA.1 is not very protective against the BA.4. So basically what you are trying to say is that infection by one variant might not really provide protection with another subvariant. But although scientists are looking into this, but then this may explain the current surge in the number of cases in Ghana in terms of the BA4 lineage. And so this is an alarm to allow for more sequencing and detection of these new variants. And in summary, basically what we've seen is that in Ghana we had more than 50 unique lineages circulating since March 2020, but then 7 of these variants of concern, such as the Alpha, Beta, Delta, Eta, and Omicron, represent almost 71% of the sequenced genomes to date. And although initially B.1 lineage, dominated local transmission of infections, but the variants of concern have taken over. And we just want to point out that the B.1.1318 variant was a variant of interest in Ghana. And basically, there's been a lot of significant diversity in terms of variants of concern that are circulating in Ghana. And this is basically because the virus is constantly evolving, and there's generation of new mutations, and you can see there's a lot of variability in terms of the number of mutations that are contained within one particular variant of concern. And right now, Omicron is leading the way with up to almost 60 mutations. And this is, a call for alarm or concern. And some of these mutations are basically unique to Ghana, and they just indicate the local evolution and suggest a significant amount of community transmission of the virus. So there's need for continuous sequencing to monitor the spread of these emerging Omicron variants, including the BA.4 and the BA.5, so that we can inform disease control measures to the Ghana Health Service. I would like to acknowledge all our funders, including the Rockefeller Foundation, the FCDO, the UKA, the ATIC, and all the different COVID-19 teams across Ghana, the different labs, our partner institutions in Nigeria and Burkina Faso, as well as all the testing laboratories that have contributed the samples in Ghana. All these different facilities, we've received samples from them, and we would like to acknowledge them for their contribution. Thank you very much.
Good, thank you very much for this. Great, so we're going to take the last presentation. Last presentation is coming from our colleagues as SJ, and will be led by Dr. Ido Olawi. So please share your screen and take it away.
Good afternoon, everyone.
Good afternoon, Prof. Oh, are you the one starting?
Yes, I'm starting. So.
Okay, all right. So Professor Folarin is the Deputy Center Leader for SG, then she's going to start. All right.
Yes, thank you, Prof. While you do shares his screen, good afternoon, everyone. And I just want to also join my voice to welcome everybody to this webinar where we are sharing our results and our sequence data. Idowu? Can we get a permit to share our screen? He's unable to share his screen right now. Please permit Idowu to share his screen. And he is also muted, so he needs to be on.
Tio, please, are you tuned in? Please make the two of them co-hosts so they can share.
Yes, Prof. Please, I have Emily Silamata Badong on the call.
No, Dr. Ido Olawoye, please.
Yes.
On the shadow, look on the shadow and go by the names on the shadow, please.
His ID is Ido ID.
Yes.
We do, okay. You may share your screen now, please.
Okay, Don, can you please share your screen?
Hi, good afternoon, everyone.
So just to start, I think our work is, our own presentation has been made a little bit easier following the two presentations. And we just want to just give us a little background about SGIP. Next slide, please. So SGIP also is like a sister institution with WACCBIP, and it was founded also in 2014 by the director, Professor Christian Happy, following the funding from World Bank. And since then, we have received support and funding from other funding agencies that have kept us going all the way. And our main focus is, the purpose of the creation of SGIP was to address the knowledge and technical gap in genomics as it affects especially infectious diseases in Nigeria and other African countries. So we're based in Redeemers University in Ede, Osun State, and some of the center leaders of the World Bank Centers of Excellence have visited us on our site. So we have partners from different African countries, but academic, clinical, and research institutions from Nigeria, Liberia, Sierra Leone, and Senegal. Then we also partner with international partners from Harvard University, Tulane University, University of Cambridge, and many others that you would see on our acknowledgement slide. We also partner with industries such as Illumina, who actually are supporting us in terms of sequencing kits, and the DMAGI supporting us for creating a database, and then also some public health agencies like the Nigerian Center for Disease Control, National Primary Health Care Development Agency, Africa CDC, WHO, and many others. Next slide. Just to know about a few of our achievements, we've since 2014, following our establishment, our true test of excellence actually came when Ebola, when we had Ebola outbreak across the West Africa region, and we tested and also sequenced the first Ebola virus sequence, and we immediately deposited the sequence data to GSAID for secondary data use. And then in 2015, we also sequenced the largest database of Lassa virus genome. And that's where we were able to uncover the origin of Lassa across the continent. We also developed rapid diagnostic test kits for the detection of Ebola and Lassa virus. And this actually got US FDA and WHO approval for emergency use. And in 2015 as well, we discovered 2 novel rhabdovirus in healthy individuals. And then these were named after the community in which they were detected. In 2017, when there was an outbreak of monkeypox in Nigeria, we also used our facilities to actually confirm the virus to be monkeypox. And I think yesterday we got a call also from one of the states in Nigeria that they have some suspected cases, and they are planning to send a sample for us to be able to also confirm if it's actually monkeypox or something else. We also used the metagenomics analysis for the detection of yellow fever in Nigeria in 2018. And then also we detected enterovirus B in an infant in Lagos State. Next slide, please. So I know the focus of our presentation today is about SARS-CoV-2, and I'm just going to skip to say the fact that SGID actually sequenced the first SARS-CoV-2 in the country and also in Africa. We've actually submitted that also on GISAID. We sequenced on March 1st, 2020, which was the first case that came into the country. And currently, Nigeria recorded 256,000 new cases of COVID-19, and this has resulted in 3,143 deaths. Just as a similar case as was presented by Professor Wandari and Dr. Kwashe, Nigeria also have recorded 4 waves during the course of the pandemic. Next slide, please. And this also is just to show us the pattern of the wave, which is very close, similar to what we have in the other 2 African countries. I'm going to hand over now to Idowu, who is a bioinformatician, to go on with the sequencing data. Thank you. Idowu, over to you.
Thank you, Professor Ryan, for the introduction and giving us an overview of the situation of Nigeria and how it has been a focal point in contributing to the.
Please, can you speak up a little bit? Sorry.
Oh, okay. So I was just.
Or get closer to the microphone.
Now, can you hear me better now?
Still quite low. I don't know why your voice is not as clear as Prof's. You are sitting far from the microphone.
Is it better now?
Hello?
Hello?
Yeah, it's better.
Okay, so thanks, Professor Ryan, for giving the introductory talk about how ESG has contributed to the pandemic, and even prior to the pandemic, how we've been a focal point to infectious diseases in the region. So I'm just going to carry on to talk about the genomic epidemiology and surveillance that ESG has carried out during the pandemic in Nigeria and some neighboring countries within the region also. So just like Professor Ryan mentioned, ESG sequenced the first case of SARS-CoV-2 in Nigeria, and which also happened to be the first case in Africa. Sorry, not the first case, but the first genome from Africa. And from the analysis that we did on that sequence, what we did realize that the sequence actually did cluster with some of the European sequences right here, which also, correlates with the travel history of the person who arrived in Lagos in February 2020. And we also made this information public. We released the genome to GISAID. We did a biological post, which, we explained how we carried out the procedure and the analysis that was done to, generate the sequences, so that other labs can also use the information to prepare themselves for whatever case, SARS-CoV-2 related case they have in the country. So throughout the pandemic, we've been doing a lot of work sequencing, receiving samples, processing them, and making our results open to the public and health authorities. And ESGAID is situated in Redeemer University, which is in the southwestern part of Nigeria. However, we do have partners all across the country where we do get in samples, and then we usually test them with real-time PCR to confirm if they're positive or not, and then we carry on to the sequencing on the samples. So initially we started with the metagenomic sequencing protocol, and then we started to adapt the American-based protocol, which is widely used in various labs across the world. So prior to the Rockefeller project, we have been able to sequence over 1,500 samples, and we've also assembled over 700 genomes from 25 states within the country. And this plot just shows the transition of the variants that we've detected over time. So just like in the older places, we'd find the parent lineages like the B1, B11, and A, which contributed to the first wave in Nigeria. And then towards the end of 2020, we start to see the introduction of variants of concern, such as the B117 and B1525. And interestingly, we did not see a lot of B117 in Nigeria. This was very much dominated by B1525, which is also known as the Eta variant, during the second wave. And also there were a lot of hypotheses that this lineage actually originated from Nigeria. And we did some analysis on this variant actually, and which is currently being reviewed in Nature Microbiology. Hopefully we'll get that published soon. And from the Bayesian analysis which we did, we were able to confirm that this lineage actually originates from Nigeria. And towards the beginning of April, we start to see, Delta lineage creep into the country, and that started to rise and outcompete the Eta variant. And this is just a map showing the countries which we've received samples from during the Rockefeller project. There are about 19 states out of 36 states and the Federal Capital Territory. And this map just shows the region where we've gotten samples from across the country. And if you notice, most of the regions are in the southwestern zone where our lab is situated. This is Osun State here. So we have a network across the country, but it's easier to get samples in from neighboring states, which is reflected in this map here. And on the right side is just a bar that just shows the numbers of states and the proportions of samples, or genomes rather, that we've sequenced from these states, with Edo State being the highest, followed by Abuja, the Central Capital Territory, and some other states from the southwestern zone. You can see all your states here, which is next to Osun State, and River State, which is in the south-south region. Also, we've gotten samples from the northern part of the country, especially Kano State. We have some samples from Adamawa, but they're low, from Sokoto, which is very few as well. And from the Middle Belt area, such as Kwara State, Nasarawa, and I think, yeah. So we tried as much as possible to try and represent all the geopolitical zones in the country and through our sampling processes. And during this process, we generated about 2,700 genomes from and over 3,000 samples that were collected. And during the Rockefeller project, also the lineage that were dominating in the country were a bit different from what we saw before the project, where we have the parent lineage and then the variants of concern such as B117 and B1525. During this period of time, that's from April last year to April this year, we do see the Delta lineage just dominates thoroughly, and then towards the end of last year, we start to see the Omicron variant as BA1 and BA11, which all merged together as BA1 here, start to rise from September. However, if you do remember correctly, this lineage was first identified in November in South Africa, and then when everyone started to do genomic surveillance properly, we also did try to sample, states within the country, and then we did find that this Omicron has actually been in the country prior to 2 months before it was actually detected in South Africa, sometime in September thereabout. And this is just a proportion of the variants in the country. That's during the period of the project where you'd see it was majorly dominated by Delta variant and followed by the Omicron variants as well. So in many states, you'd find purely green colors there, which represent Delta and Delta sublineages, and then followed by the red color, which is the Omicron variant. So this is just a brief, introduction to what the Delta is. This cessation of Omicron variant in the country. Just like I said, the first case was detected in Abuja on the 25th of November 2021. However, we've now found that this was actually in the country 2 months prior to the time. Now, the first case was identified. And then currently we found about 600 samples out of 14 states that were, sampled, and all Delta variants. And these are like more recent samples. And these have characteristic mutations in the spike protein, which we can see here. Delta variant has this highly mutated spike region where it's gone under environmental and host pressure, and this is what we find in recent samples in the country. So talking about the Delta variant, which has the highest number of variants so far in the country. This is about over 2,000 samples, literally, and from all the states that we've sequenced from, we've had to find Delta in all these states. And the first sample was from Ondo State, which was collected on the 20th of May, 2021, and this also has several other lineages, such as the AY42, AY36, and AY102, and many others as such. However, in Nigeria, what we did notice was that the AY36 variant seems to be the prominent Delta sublineage in the country. At the time, we did have the AY109, but it didn't last as much as the AY36, which I think is the same situation with Ghana, if I'm correct. But this seemed to be, what dominated amongst the Delta sublineage until the Omicron variants came to, outcompete it. So as far as the Nigerian genomic surveillance, we've sequenced samples from Burkina Faso and Rwanda during this period of time, and we've assembled about 160 genomes from Burkina Faso, which majority are not Delta or variants of concerns. I think these samples were sampled during the early period of Delta variants, so we didn't grab the actual situation of the third wave in that community, and about 30% of them were Delta variants, and we do have some of the Eta variants in that locality as well. And from Rwanda, where we have about 170 genomes sequenced, we do have majority of them were all Delta variants, and then we do have some Beta variants also. So what does this mean in the public health side of things? Well, we do intend to carry on doing more genomic surveillance, because just like our first presenter did mention, there seems to be a sudden surge in cases now in Africa, which might be as a result of the BA5 and BA4. So we don't intend to let our guard down. We intend to carry on active surveillance within our country and also neighboring African countries where we can get samples from to know what the situation of the pandemic is in our region. And we do also notice something, which is like a resurgence of Delta variant at one point in time, but it's went down again, and we're not quite sure why that could have happened. And so, which is why it's important to keep carrying out genomic surveillance and make sure it's a continued process, not just a one-off time thing. So I would pass it over to Professor Florian to conclude the presentation. Thank you.
Thank you very much, Idowu, for that presentation. So I just want to round up to say, what have we done with the sequence data that we have been able to generate? All the sequence data were submitted to GSAID for secondary data use, such as drug target discovery, vaccine candidate discovery, and also better diagnostic tools. As we know now that it's real-time PCR that is still being used, but there's a need to see how we can design and develop a rapid and accurate diagnostic tool that would enable prompt detection of this virus. And then in addition to depositing all the data, all the sequence data on GSAID, we also communicate the interpretation of the sequence real time to the Nigerian Center for Disease Control and also the Presidential Task Force for COVID-19. And all of this information have been useful in policy towards the management and control of the disease in the country. As said by Idowu, the dominant variant of concern in Nigeria is currently Omicron. And just like it was presented also in Ghana, we also observe evidence of community transmission in different states of the country. All the mutations that have been reported on the spike proteins are of a major concern to us because as they keep accumulating, since the primers were designed around this region, they may also lead to false negative results at the end of the day. And so there's a need to just keep monitoring and be able to check whenever we are having such a case, just to be sure that we're not having false negative results, because these individuals will go back into the community and they keep spreading the disease. Next slide, please. We also would like to, next slide. These are the beautiful faces of all our team members during the peak of COVID-19 and where they actually carried out all of the sequences. And we also like to acknowledge all our partners, including the Redeemers University, as well as all our funders. Next slide. All our partners and funders, especially Rockefeller and the FDA and many others, World Bank, and many of all our partners, as you can see. Thank you very much. Over to you, Professor Awandere.
All right, thank you very much. So let me attempt to summarize and then we'll take the discussion. So for the genomics work, we see that there were clear variants of concern driving each of the waves on the continent, even though the waves in Africa seem to lag behind those of the world, except for South Africa. South Africa is always ahead of the rest of Africa. West Africa seemed to lag behind the rest of the world in terms of when we had our surges. And each surge was driven by one particular dominant variant. Currently, we are in the Omicron era and Omicron is dominating. We've seen that the subvariants of Omicron are also in competition. And it is interesting to note that we've now detected BA4 and BA5 in Ghana for the first time. So this is a significant milestone because it probably explains the increasing number of cases we're seeing. And it means that we have to get back to some of the protocols that we have lacked because these two variants, as my colleagues mentioned, the protection from previous exposure to the Omicron BA1 and BA2 does not protect adequately against these two variants if you are not fully vaccinated. So it's important that people get fully vaccinated in order to protect them from the new variants. Our colleagues from Nigeria have not yet reported BA4 and BA5. I'm sure in their next few weeks they may find some of them. It was interesting to note that there was a re-emergence of Delta a little bit in December and then disappeared. That's something that's interesting and we should be monitoring. Altogether, the seroprevalence, I think the message from the seroprevalence is that COVID has been here and it has spread quite a bit because of if you look at the levels of people with antibodies, remember that most of these kits that we use for the work, they were detecting the nucleocapsid, which is not the target of the vaccines. So these people were mostly naturally infected, these antibodies that were picking up. So this basically shows that the disease has spread quite a bit across West Africa. We have been fortunate that it hasn't been severe due to steps that various ministries of health and the Ghana Health Service and other health agencies have put in place to minimize the impact of the infections, but the infections have occurred. And you can see that in Nigeria, Burkina Faso, you find the same thing. We've seen studies from Malawi, South Africa, we find the same high levels of antibody prevalence. So I think that we should minimize the backslapping and saying that Africa somehow we have some magic bullets that we use to control COVID. No, we got infected all right, just that it wasn't severe. I think where we need to focus our attention now is to understand why it wasn't severe. And I think that is how we can contribute to scientific knowledge, because I think there's something unique about Sub-Saharan Africa, that despite such high levels of exposure, we haven't had that many deaths. There have been studies trying to claim that there were excess deaths and that there were unreported deaths and all that. I think that I don't buy all that. The evidence is very weak. I don't think we lost that many people. It's as simple as that. We should try to understand why the disease wasn't severe here, but we shouldn't deny that it wasn't severe and that people have died, but we haven't accounted for them. So I think we shouldn't buy that type of analysis, which mostly comes from colleagues who are not here, who are in Europe and the USA and sitting behind computers and modeling. So anyway, I'm going to call on the other discussers who have not presented so they can give their comments. Dr. Yabedi Aku, CEO of Yamachi is here. He's a research fellow and an entrepreneur. If you have some quick comments.
Thank you, Prof. So I think you actually captured most of my comments in your summary. I think it is clearly, well, first of all, congrats to all the teams involved. I think it's great that this work has been done in Africa. If maybe a few years ago, this may not have been possible. So I think it speaks to the capacity development that has been built at centers like WACCBIP and ACEGID and others. But I think if we look at the data, I agree with you entirely that we cannot say that COVID has skipped Africa. In fact, COVID is still very much present. If you look at the most recent data that Dr. Kwashi showed, seroprevalence rates, seropositivity of over 70, close to 80%, means 8 out of 10 people have been within the past probably 3 to 6 months exposed to the virus. So I think we still, we cannot say that outbreak is over by any stretch of imagination. Likely the case numbers we've seen declining were probably due to antibody levels that were minimizing infections. Once travel restrictions were withdrawn, majority of testing in our countries also declined. So we're actually not detecting anywhere near the number of people who are actually COVID positive. I think what we are seeing now with this new quote unquote surge is BA4 and 5, which as I think Collins mentioned, has been shown not to really, is a sort of immune evasive relative to people naturally exposed to the first versions of Omicron. And I think you can see, based anecdotally at least, these infections are a little bit more severe in that people are at least feeling ill enough to want to get tested. I can say that even from here at Yamachi, where we do COVID testing, we are seeing greater numbers of people coming in to do a test. And these are not people who are traveling. These are people who are perhaps not feeling very well and want to know what they have. So I think because BA4 and 5 are manifesting slightly differently, are less asymptomatic, more and more people are choosing to test. And I think that is why we are now picking up more and more cases. The final thing I'll say is it also emphasizes the need for vaccination. There is very good data that shows that a vaccine plus a natural exposure can actually protect against BA4-5 better than just natural exposure. And so in many African countries where our full vaccination coverage is still south of 20 or 30%, we need to do something about that. And I think now we need to start talking about boosting, because now the definition for fully vaccinated is no longer sufficient to say you've received 2 shots at some point in your life. It is now you are within 6 months of your last COVID shot. And so the definition for fully vaccinated in Europe now, if you're traveling to Europe, you need to have a vaccine within 6 months. You require a booster. And I think that is in response to the science. So I think something to discuss now is, given the resurgence, given the fact that 4 and 5 are making people at least to some extent symptomatic, do we need to push for more use of boosters in our country since the vaccines are currently available? And do we need to change the definition of fully vaccinated to now include the fact that you need to be 6 months, within 6 months of your last COVID shot? So those will be my comments for now. Thank you very much, Prof.
All right, thank you very much. Professor Sodio Monsirma, do you want to comment?
Let me just activate my microphone. Thanks a lot for giving that opportunity to talk. First of all, let me thank those who have presented and also congratulate you for being able to gather so many people around and conducting what has been done as study. So regarding the result, I think in Burkina Faso here, we just have been very limited in terms of providing material to you. What we have sent was just material from up to, let's say, September, meaning that our first initial round didn't cover the Omicron wave. That's why during your first presentation related to 2021 data, nothing was shown in terms of information for Burkina Faso. And all material sent for sequencing was sent in 21. Then Omicron wave was not yet occurring. That's why maybe people have seen that.
Prof, your microphone has gone low. I don't know why. We can't
hear
you.
Really? Are you getting me?
It's gone faint a little bit.
Okay, so if so, what I can do is just change the microphone. Just give me a second. I will change microphone and remove the.
Okay, let me come back to you later. I'll move to others and I'll come back to you later. Dr. Issa Aliyu Abubakar from ASFAB, Bayero University. You want to comment?
Yes, good afternoon, everyone. Actually, what I wanted to comment has been discussed in detail, but I still want to commend the presenters, the Takashi and then other presenters.
Your voice is very low. Can you speak up or get closer to the microphone?
Okay.
Okay, go ahead. Yeah.
Can you hear me now?
Yeah.
Actually, all that I wanted to say has been discussed in detail, but I just wanted to add some few comments. The results from across 3 countries shows that what we've been seeing is like an ice patch. What has been reported as the COVID-19 prevalence in Africa is actually far beyond that. We've seen in some sites more than 80% of individuals screened were exposed to the COVID-19. And then the issue of the COVID-19, in Africa, we doesn't have symptoms of COVID-19. It's actually something to actually, we need to have a detailed scientific evidence to found that, because in most of the African countries, the cases where undetected people are dying, and we may not probably know the cause of the death, and so on and so forth. So actually, the results of this study actually warrant often those for more research to actually establish the actual COVID-19 cases in Africa, and then what extent of the disease burden in Africa. And then for the testing we do, if we tested for the RDT kit we use, if we use the kits, we use kits that detected COVID-19 nucleocapsid protein. This will detect individuals who have exposure to natural infections. And then, but if somebody is vaccinated, the vaccines approved by FDA and for emergency uses available in the world are actually targeting RBD of the spike protein. So when individual is vaccinated, the M protein tends to disappear. And then if you use only N protein targeted kits, probably you might be missing some of the cases. So probably the site that only use N protein targeted kits only, the actual result might be a bit higher than what is being reported, because individuals who are vaccinated might probably not show the N protein antibodies. So in essence, these are the picometer, but in most sites, like in our site, we actually use both the kit that detected both the antibodies of the nucleocapsid protein and the RBD protein. So you may be seeing the actual exposure level and then the vaccine-induced antibody and individuals. I think I say thank you very much for the opportunity. And
Thank you very much. Earlier on, we had Ghana's presidential advisor on health here, but I think he's dropped. I can't see him over there. Dr. Isiasa, are you still on? If you're on, if you want to comment, I saw him on earlier, but maybe he has left. Our National Laboratory Testing Coordinator is also here, Professor William Ampofo. Please, if you want to speak, you have some comments on flu and COVID.
Yes, thank you very much, Gordon. I'd like to congratulate WACCBIP and the other centers for this excellent seminar this time, because it's really great to show what we African scientists, especially in the sub-regional, are capable of doing. So I think it's important to note that for us in Ghana, because we do surveillance of flu alongside COVID, we have also observed some increase in flu cases over the past month in May. Now, this is interesting because in the tropical climate that we have, we do have flu all year round, and we have observed that we do get some increases of seasonal influenza at the beginning of the rainy season. And it's interesting that throughout the COVID pandemic, we have continued to see flu cases, albeit on a lower level in Ghana. So I would encourage that, genomic surveillance of SARS-CoV-2 also looks at other respiratory viruses, especially influenza, because for some time now we've been discussing the possibility of including doing the case bedding, the disease bedding for influenza, and also introducing, making a case for the introduction of influenza into the routine immunization. So now that we have discussions on COVID immunization, it will be important to look at flu alongside it, because the US CDC and WHO have incorporated surveillance for flu alongside COVID-19. So this is a point I wanted to make up. And very interestingly, when we had a lot of Omicron, in Ghana recently, we had very little flu. When Omicron went down, we had an upsurge of flu, and there seems to be no cross protection from the data that we have seen between, the Omicron strain and the flu outbreaks in the close settings that we've seen so far. So I think it's very interesting to look forward, to going forward to look at, these other pathogens as well. Thank you very much, Chair.
Thank you, sir. Very, very, very important. So I think, the point is a very important one, to incorporate basically flu surveillance, as well as the sequencing, genome sequencing or genomic surveillance of flu as well. But I'll bet that one of the reasons why the flu went down was that whenever the COVID cases are high, I think because of the protocols we observe, people washing their hands, sanitizing, wearing masks, social distancing, almost all diseases go down because the hygiene just goes up. So it may be one of those things that is just a byproduct of increased hygiene. And then when the cases go down, we all relax and start putting the mask away and shaking hands. Over the last few weeks, everybody's shaking hands now because we thought COVID had gone. But as you can see, COVID is not gone, it's coming back. So it could be one of the reasons. So very, very important. And now flu and COVID, I think we're going to be dealing with these two for quite a while. So we have to have a joint surveillance, vaccination, genomic surveillance for the two as well. All right, thank you. If anybody else has a comment, you can raise your hand. I would call you. We still have about 10 minutes. But I want to go back to Peter. Peter is the overall coordinator of this work. He helps me with managing this enormous project across several countries. So Peter, do you want to come back with any comments or questions that you have picked up from the chat that you want to address?
Yeah, so I think I'll start off by trying to address some of the questions that have been in the chat. The question is, I think the first one is, what must have accounted for the rise of Omicron and continues to reign? And I think there's something that we've touched on earlier, that Omicron was one that was first detected in South Africa, but it looks like it was earlier in Nigeria. And the rise is basically probably due to, or the rise that we're seeing now, at least in our context, as Prof just touched on, is probably due to relaxing of the protocols as we saw the drop in cases. In Ghana, for probably the last 2-3 months, we haven't really seen many cases in Ghana. I will address. There was another question. Are we on the verge of experiencing another wave in Africa? I wouldn't go as far as to say are we on the verge of another wave, but it looks like we might be. So it's too early to say that now. Time will tell whether we do have some protection or whether it's severe or it's not severe. Have we done any work with wastewater? There are some colleagues which are at least not WACCBIP, not yet. It's something we've had discussions about. And I'm just trying to go through all the questions. The question is, did any of the variants like Delta confer protection to later variants? So Collins touched a little bit on this, that it looks like yes and no. We didn't know whether the reason why Delta was not so severe was because of prior variants, but it's also likely and has been reported in South Africa that the BA 1.1 doesn't confer resistance to the BA4, BA5, but it might have conferred a little resistance to BA2 and other Omicron subvariants. What's interesting is this was also mentioned earlier, is that vaccination actually, having a vaccination plus your natural infection is what really seems to give you that added level of protection. And I think some of the analysis we had done earlier, we had seen that people who were vaccinated and who had natural infection were less likely to have symptomatic infection. So what we are seeing now with BA4, BA5, which seem to be more symptomatic infections, is probably because most of these guys, if you look at the vaccination data, have not been vaccinated. They likely were infected with Delta or Omicron. And now they're getting infected with, we think, BA4 or BA5. We think maybe BA4. And now they're showing a bit of symptoms. So time will tell how severe that will be. Let me see. Have we been able to link RDT performance to any variant? As has been mentioned, most of the RDTs that we are using, so the one we used in Ghana and Burkina Faso is nucleocapsid based. So it would pick up any variant of spike. In Nigeria, it was SD biosensor, which is nucleocapsid and spike based. So it might have some performance issues with some variants, but we did see increasing cases, increasing seropositivity. So there's no evidence so far that its performance was affected. Yeah, so I think that's okay. One question I'm seeing. My concern is, do the centers across West Africa have surveillance mechanism for early detection of potential virus infections that could originate locally, like working with health centers? So that's something that we are doing, and I know all our partners are doing, and I can speak to WACCBIP here. We've tried to build a very strong network with the primary health facilities, with Ghana Health Service, with some of the other health networks in the country. And so once strange things happen or strange diseases are showing up, we usually get in touch, they get in touch with us, and then we try to do the investigation. So let me just see if there's any more questions. We are not just assuming all these variants are important. I think that's a question. I think Collins presented some molecular dating analysis. Of course, we can't go into too much detail in this forum based on time, but we are not just assuming all variants are imported. What we didn't present is that early on, we always detected the variant at the airport before you detect it locally. I don't think there was ever any instance of a variant, apart from the A lineages, where we detected it at the airport before we detected it locally. And then again, you can tell post-Omicron, when in January, February, when we had the peak and the cases died, this is sort of something, the rise of 4 came after BA4 and BA5 were already causing surges in other countries. So it's unlikely that this is something that was evolved locally.
All right, thank you. So there's a question here about cross-reactivity, which is something we should talk about. So we didn't have time to explain, but the way we chose these kits that we used to do the work was that we used them on, convalescent plasma from people who had recovered from COVID, and also negative controls from samples we had collected, long ago, pre-2019 samples, okay? And we use that to rate the performance of these kits. So we did this together with Noguchi. Some of these were done here, some were done in Noguchi. But if you put it all together, you were able to choose those that were the best performing in terms of discriminating between real positives and any false positives, such as picking up positives in samples that were collected in 2017 or 2018 when we didn't have SARS-CoV-2. So the idea about cross-reactivity we've seen some publications talk about cross-reactivity, but these are method specific. It depends on what method you are using. If you don't do the necessary controls, of course you have some cross-reactivity. If your method is not working well, you pick up all kinds of stuff. But we took time to select the kits to do the proper quality control before we went to the field. And so we are very confident in what we're seeing. And like others have mentioned, if you look at the trend, the way it matches with the waves, that is not Ebola or yellow fever or anything like that. What kind of yellow fever cases rose from 2020 to 2021 to 2022? If you look at the trend of our positivity rates. So I really don't think that we're talking about cross-reactivity. We've done all the proper controls for that. All right. So I think our time is up. We are very grateful to all of you for making the time. I think that this has been very interesting and very interactive, and the attendance has been very, very impressive. At some point, we were almost at 300 people from across the world. So that's great. We're glad that people are interested. You can continue to follow us on Twitter, Facebook, and on our website, which is WACCBIP.org. All our work we update on our social media pages and then on our website as well. So you can follow us on Twitter and Facebook and whichever social media platform you want. Going forward, we're going to continue to do this work. We have support from FCDO to continue to do this. We have support from Wellcome Trust together with Arctic and then University of Cambridge to build capacity in West and Central Africa. So we're going to be training labs and standing them up to do mineral sequencing in West and Central Africa. So if you have the basic capacity and you are interested, get in touch with us. We would include you in our future training so that you can be able to do mineral sequencing. The idea is to, basically empower as many labs as possible